Welcome to Hephzibah Simple Class. Today, I will be talking on Arabinose Operon. To start with, what is an operon? An operon is a unit of genes which are under the control of a single promoter where RNA polymerase binds. An operon functions in a coordinated manner by means of the structural genes, promoter, operator, and some other units. Now, what is an Arabinose operon? An Arabinose operon, also known as Ara operon, is a unit of genes that encode enzymes responsible for the catabolism of L-arabinose sugar to deoxyxylose 5-phosphate, an intermediate of the pentose phosphate pathway. Similarly, Arabinose operon is a sequence of genes and other regions, all classified as structural genes, promoter, control sites, and regulatory gene. However, an arabinose is an aldopentose, a 5-carbon atoms monosaccharide sugar having an aldehyde functional group. The L form of arabinose is more common in nature than its D form. L-arabinose is found as a natural constituent of sugar beet and most fruits and vegetables, as polysaccharide or biopolymer like hemicellulose and pectin. Although Escherichia coli consume glucose before any other sugar or nutrient as their source of carbon. When arabinose is present in the environment, Escherichia coli take it up, especially when they are starved of glucose. Note that L-arabinose enters Escherichia coli either by a low-affinity proton-driven transporter, designated as ARA-E, or high-affinity ATP-driven system, designated as ARA-F, ARA-G, and ARA-H. On the Arabinose operon, the structural genes are togetherly transcribed into a messenger RNA strand, which may be translated or split to create monocistronic messenger RNA strands that are translated separately into proteins. This simply means that the genes contained in Arabinose operon are either expressed togetherly or not. What are the structural genes? The structural genes encode enzymes for the L-arabinose catabolism. ARA-A encodes L-arabinose isomerase, which catalyzes the conversion or isomerization of the substrate L-arabinose to L-ribulose. ARA-B encodes L-ribulokinase, which catalyzes the phosphorylation of L-ribulose to L-ribulose 5-phosphate. ARA-D encodes L-ribulose 5-phosphate for epimerase, which catalyzes the conversion or epimerization of L-ribulose 5-phosphate to deoxyxylose 5-phosphate. Now, what does a promoter mean? A promoter is a sequence of DNA to which proteins bind and transcription of DNA to RNA is initiated. Now, let's see what control sites are. The control sites are DNA binding sites that, when occupied by regulatory gene products, they induce expression. What is a regulatory gene? A regulatory gene is located upstream the Arabinose operon. The regulatory gene encodes Arabinose responsive regulatory protein, also known as regulatory gene products. a dimer protein with two binding domains. Notes: the regulatory gene and the structural genes are transcribed in opposite directions from the regulatory gene promoter and structural gene promoter respectively. In conclusion, there is also a catabolite activator protein site on the Arabinose operon, where catabolite activator protein cyclic AMP complex binds to facilitate catabolite expression, which subsequently results in a positive regulation of the structural genes when Escherichia coli are starved of glucose. This catabolite activator site is situated between the control sites. Here I come to the end of this class. I shall talk on regulation of Arabinose operon and catabolite repression in our next class. I want to acknowledge and appreciate owners and authors of every image and material used in this class. Please always remember to subscribe to this channel. Thank you.